form of a mass spectra. This can be done for around one peptide can be determined in 10 to 20 minutes. So you need bioinformatics help. This is not possible by humans. So what you do is whatever happens inside the mass spectrometer, that phenomenon, you create in silico from a protein database. You take a mass protein extract from the sample, you take the protein database, computationally you do whatever happens in the mass spec through computational algorithms, and you create all possible theoretical mass spectra from a protein database, and then you compare experimentally derived mass spectra with theoretically derived mass spectra. You tell that this peptide is identified with giving statistical parameters and quality control, many other things. So then finally peptides you map back to the proteome and you tell them this protein is identified based on these peptide identifications. So now what happens is the experimentally derived proteome is not fully represented in the protein database. You miss all the novel proteins. You miss all the proteins which are modified. So for that purpose, you can use proteins database of closely related species. You can even translate genome. If genome is available, you translate it into 6 frame, and or you can use RNA-seq data sets and translate them into 3 frame and create protein databases, conceptual protein databases, which can be searched. The protein what you have extracted can be searched against that. Then if some proteins are coming exclusively from these databases you search, which is not represented in the protein database, then it is the time to add that protein into that database. You identified novel proteins. So some peptides, collection of peptides falls into a genome area where there is no protein is defined, you identified a novel protein. If it falls into an area where half the exon is represented but it extends, you are modifying that exon structure, because protein is real. Gene structure is not real because it is computationally derived, most of the genes. Okay, we have not seen the proteins. All proteins we have not seen today. All the proteins which are there in the database are conceptually derived from the genome through computational approaches. So when you detect a protein, peptide, from the mass spectra, and then it is reasonable to tell that this is the real structure of the protein, and you redraw protein annotation. This is called proteome driven genome annotation. So we have done this for several organs of human for the first time, and it appeared on the cover of Nature, and this is the first protein draft map of human proteome, and 90% of the authors here are from Bangalore. So we Indians never participated in the draft map of human genome, but we Indians led draft map of human proteome for the world. So we also did subsequently for the same exercise for human brain, different parts of human brain. All these are Indian brains, not Caucasian brains. So you have a first draft map of human proteome map ready now. We have to submit it to, click the button to submit this manuscript now. So this is for protein identity for you. Now we will say how the protein expression is different across the tissues. So we have methodologies in mass spec. You have labeled methodology and label-free methodologies, SciLab, ITLab, TMT, several methods. So with which you can determine proteins which are changing, and then what is the proteins which are modified by adding several functional groups on specific proteome. So one of them is phosphorylation, and which is mostly implicated in several signaling pathways and all. So we have there are methods to isolate or enrich these phosphoproteins and then evaluate. So now you apply the phosphoproteomics method with the quantitative phosphoproteomics, then you can differentiate between not the expression which is happening in the transcriptomics methods or Western blot or other methods. You are identifying proteins which are different. It is not that expression wise difference, but only in the proteins which are phosphorylation differences. So this happens, you can identify phosphorylations which are changing, not the protein expression which is changing. which is more functional because it gives activity to the protein. So now we complicate it further. What are the phosphorylations which are changing in 2 minutes, 4 minutes, 10 minutes, 20 minutes? When a drug is given or an insult is given, or which results in the functional changes. So this is time-lapse quantitative phosphoproteomics for you, with which you can derive, and if you develop proper animal models and cell line models, you can derive what is happening instantly when a drug is given or when a disease is modified to recovery model. So whether disease model, whatever phosphorylation changes happening, are all recovered by the drug during a recovery model. So this will give you a lot of novel drug targets which can change epigenetics, then it can change transcriptomics and bring about the change from disease to recovery. This is where we are specialized in. Finally, you can map these phosphorylation changes across this 2 minutes, 4 minutes, 10 minutes, 20 minutes. From here, transcription factors and epigenetics will start. So this is regulation of entire epigenetics, what is happening with the drug. So this is very important. These are all available, accessible very easily outside the cell. So most of future drug targets which we have to develop lie within these 2 minutes what is changing in any drug is given. So now shifting the gear to